Now here what we're going to look at is the general concepts behind addition-elimination reactions. We'll use hydrolysis of amides as the example, but really all the patterns here are observed in other acyl compounds as well. But let's talk about the specificity of amides and what makes them different. It turns out hydrolyzing them is not the easiest task. We need to use strong bases or strong acids and introduce significant amounts of heat. So here we have benzamide, and we're doing a base hydrolysis here with heat to form sodium benzoate and ammonia. So essentially what we're doing is transforming the amide into the carboxyl, rather carboxylic acid, or carboxylate in this case, and the free amine, which may or may not be protonated, depending on the conditions. So in DMSO, ammonia has a very high pKa of 41, and when attached to carbonyls to form the amide, it drastically changes the behavior of this acyl compound compared to esters, carboxylic acids, and more extremely compared to acyl halides. Because this amine nitrogen, which is bound in the amide, is able to share its electrons to form a pi bond here. And because this lone pair is tied up in this resonance structure here, we can consider this amine a lot more acidic compared to ammonia, which doesn't have any of this resonance. So that's why benzamide, in relation to these hydrogens here, is about 10 to the power of 18 times more acidic than ammonia is, if you just compare the pKa's in DMSO. So it's this sharing of electrons in the amide that makes it such a stable compound, which is why amide linkages, also known as peptide bonds, are the backbones of all proteins. It's a very stable linkage. So now when discussing how to break that, now we know why we need heat, sometimes close to 110 degrees. So to hydrolyze in base or acid, we need heat. So theoretically, if this hydrogen is removed, we form a negative charge here and an extra lone pair. But just like we showed here, this lone pair can be involved in resonance with the carbonyl, which means this negative charge is effectively spread over a larger area right here, which contributes to stability. So now let's get into the mechanism. How do we break up amides into the carboxyl portion and the amine portion? And before we jump into it, we are using amides as the example, but the pattern here is pad-ped. If we're using acid, we need to activate the carbonyl via protonation and then carry out the addition-elimination, which is observed in esters, carboxylic acids. Whenever we're dealing with the addition of weak nucleophiles, this pad-ped pattern is observed, where first we do a protonation of the carbonyl and then carry out the addition and then finally the elimination. And scattered throughout are protonation and deprotonation steps, which essentially function to lead towards the addition and then the elimination. So these reactions are addition-eliminations, which is just another way of saying nucleophilic acyl substitution. Some sort of nucleophile is being added, which is eliminating the group that is being substituted. In acidic solution, acyl compounds require protonation when being attacked by weak nucleophiles. So the same thing occurs with this amide. Because this lone pair is tied up in resonance, it does not get protonated. So the proton is attached onto the carbonyl oxygen like so, which forms our resonance stabilized cation. Now directly after protonation, the positive charge is concentrated on the oxygen. But through resonance, what we see is that the positive charge is effectively spread over 3 different atoms, where if the carbonyl electrons move to the oxygen, we form a positive charge on the carbonyl carbon, and then the amide lone pair could come in and further spread this positive charge. So this cation is stabilized because of nitrogen's ability to donate electrons. Now the next step is the addition step, the nucleophilic addition. So because we've protonated the carbonyl, water generated in the first step acts as the nucleophile, attacks the positively charged carbocation. It attacks, the carbonyl breaks, sending electrons back up to the oxygen, and we form this charged tetrahedral intermediate. Now if we look at our patterning, the next step is deprotonation, which makes sense to form the stabilized tetrahedral intermediate. So that's exactly what happens. In the next step, another water molecule can come in and deprotonate our former nucleophile. So water comes in, plucks off the proton, and we generate this stabilized tetrahedral intermediate where we now have a hydroxyl group. Now this step is reversible because the hydroxyl can take on a proton to form water, and water as a good leaving group can jump off the compound and we're right back to step 2. Remember, the goal is to form the carboxyl group. And in this case, in acid, it's the carboxylic acid. So we need to remove this nitrogen group here, this amine group. And that's accomplished by protonation. So now we've entered the elimination stage of this mechanism. We need to protonate the amine such that it becomes a good leaving group. So with the acid generated in step 3, the amine is now protonated to form water, charged tetrahedral intermediate, which then allows for the critical step of the elimination. So the carbonyl can regenerate from any one of these OH groups, which effectively kicks off the leaving group to form ammonia gas, and what we get is this familiar structure, this protonated carbonyl, where through resonance we have a dispersion of positive charge across the carbonyl carbon and the carbonyl oxygen. That's important because, theoretically, as a reversible reaction, ammonia could come in and attack the carbonyl carbon, because it does bear a partial positive charge. But if we continue on to form the carboxylic acid, the final step is to stabilize this protonated carbonyl. It turns out this is very acidic and readily releases its proton to ammonia, which plucks it off to form the stabilized carboxylic acid and our protonated amine. Now in 6 steps, we've undergone addition-elimination, which also occurs in other acyl compounds. Now the base-promoted mechanism is a little different. The acronym here is ADPER, which means there is no activation of the carbonyl. In basic conditions, the hydroxide anion is strong enough to attack the carbonyl carbon directly. So that is where this addition step is. And as we'll see, the elimination of our amine group is actually catalyzed by the regeneration of the carbonyl, which chemically speaking is very stabilizing to reduce the delta G. And throughout the mechanism, we need to keep in mind that something needs to be substituted. We need to add something and eliminate another. So let's go through the mechanism knowing that. In step 1, the hydroxide anion directly attacks the carbonyl carbon, which sends a pair of electrons up to oxygen to form this charged intermediate here. Now this modified benzamide undergoes the next step of deprotonation. Hydroxyl loses its hydrogen, its proton, to form this kind of charged intermediate, and water. Now this prepares us for the regeneration of the carbonyl, because then any one of these lone pairs can come down and re-establish the carbonyl here. So that's the reasoning behind losing this proton, to regenerate the carbonyl with one of these lone pairs and kick off our amine group, which is not really a leaving group yet. So that's where this third step is, the protonation of our leaving group. The water generated in step 2 acts as the conjugate acid, and what it does is that the amine group gets protonated, water becomes hydroxide once again, and now we have 3 charges on our compound, which is not stable. This is just an intermediate. But more importantly, now we've formed a good leaving group, NH3. It's a protonated form of our amine. So theoretically, out of the 3 lone pairs, one of them on any one of these 2 oxygens can come down and reestablish a carbonyl, effectively kicking off the leaving group, which is the final irreversible step, the regeneration of the carbonyl and the elimination of the leaving group. And we said the regeneration of the carbonyl decreases molecular potential energy tremendously, which is why it is a favorable chemical behavior. So the carbonyl regenerates, and we kick off our leaving group. So now we form an amine and the carboxylate as a result of base-promoted hydrolysis. If we look at acid-promoted hydrolysis, we form the carboxylic acid and the protonated amine, and that's just because of the excess protons in solution, which contrasts in basic solution where we have a lack of protons and therefore we have more neutral and negative species.